Hi everyone, welcome to our presentation on how exercise promotes satellite cell proliferation.
Myogenesis describes a process of skeletal muscle regeneration involving muscle stem cells known as satellite cells. Upon stimulation, inactive satellite cells get activated and proliferate into myoblasts, which can then take two paths. One where they self-renew to replenish muscle stem cells, going back to the inactive state, while the second is differentiating into myocytes, which fuse with existing muscle fibers to increase support for muscle growth. To determine satellite cells' myogenic states, researchers had used the expression of transcription factor PAX7 and myogenic regulatory factors of MyoD and myogenin. Expression of PAX7 is used to identify quiescent satellite cells, while expression of MyoD is used to identify proliferating satellite cells, and finally myogenin is used to identify differentiating satellite cells. For satellite cells to progress through proliferation and differentiation stages, they must enter and leave the cell cycle depending on cyclin D1 and P21, which is a cyclin-dependent kinase inhibitor. Using the combined information of MyoD, myogenin, and cell cycle markers, this has allowed researchers to determine the satellite cell myogenic state. Early steps in the muscle repair process involve PAX7 plus quiescent satellite cells getting activated to transiently upregulate MyoD. When PAX7 is co-expressed with MyoD, this indicates that the satellite cells are in a proliferative state. Later steps in the muscle repair process involve a downregulation of PAX7 and MyoD, along with an upregulation of myogenin. This indicates that the satellite cells are in its differentiation state, where satellite cells can differentiate into myocytes, which then fuse with existing muscle fibers to aid in the repair of damaged muscle tissue. Other variables that can affect muscle repair include the stem cell microenvironment, which is a key variable, along with circulating growth factors and hormones.
Testosterone belongs to a class of male hormones called androgens. The ovaries in women also produce testosterone along with estrogen. These sex hormones are involved in the growth, maintenance, and repair of reproductive tissues. These hormones play a role in key differences between the sexes. Testosterone has an anabolic or building up effect on muscle growth. Resistance exercise releases anabolic hormones, which is beneficial for muscle adaptation, size, and strength. A study was done which showed that if testosterone concentration with increased number of satellite cells and myonuclei were present, there would be a greater level of enhanced differentiation and fusion. More differentiation and fusion basically means that there will be more muscle growth. Estrogen is shown to provide a protective effect against disruption of muscle structural proteins and damage-induced inflammation and support satellite cell proliferation but not differentiation. This means that the testosterone differentiation capability in men allow for enhanced muscle growth in comparison to women, yet the estrogen in women allow for a faster repair process. Men build more muscle than women, whereas women repair muscle faster than men but cannot build as much muscle.
This study included only young men and women, as older individuals have a lower rise in their circulating anabolic hormones after resistance exercise training. Researchers had found that young men have a greater number of PAX7 plus satellite cells than young women, which could attribute to the microenvironment. Eccentric exercises were also studied for men versus women, where eccentric movements involved the lowering part of an exercise, such as the lowering of a dumbbell when performing a bicep curl. It was found that untrained men had a decreased content of MyoD 8 to 72 hours after exercise, while untrained women had an increased MyoD concentration after that same amount of time. When it comes to the process of myogenesis, after satellite cell proliferation comes differentiation, where satellite cells can differentiate into myocytes to fuse with existing muscle fibers, while adding myonuclei to those existing fibers to increase support for muscle growth. Researchers had performed a 16-week resistance program, and it showed that the increased number of myonuclei was seen only in young men, as young men had an increased basal testosterone concentration before and after training.
Growth hormone promotes myoblast fusion. Cortisol is a stress hormone released by the adrenal glands. It's important for helping your body deal with stressful situations as your brain triggers its release in response to many different kinds of stress. Cortisol inhibits satellite cell proliferation and differentiation by upregulating myostatin expression, which means that the muscle will not grow. The cortisol response to weight training or resistance training is similar in men and women, yet due to the fact that more testosterone is present in men, it will suppress the catabolic effect of cortisol, thus having a greater impact on women. The purpose behind this study was to determine the effect of resistance-induced hormonal changes on the satellite cell myogenic state or the proliferation and differentiation in response to muscle damage. This study was done because there has been no prior research to this. In order to test this, they made both men and women complete unilateral eccentric knee extensions. After the knee extensions, the exercise group participated in a 20-minute upper body resistance exercise to induce a hormonal response known as EX, and seen by the black color in the figure. The other group, which was a control, rested for 20 minutes instead and is abbreviated as CON and is seen by the gray color in the figure. Before the knee extension, 12 and 24 hours after the upper body RE or rest period, muscle biopsies were obtained to measure the myogenic factors involved. In men, the data shows that the area under the curve, or AUC, of testosterone, growth hormone, and cortisol were greater in the EX group than the CON group. The area under the curve in part D of the figure shows the ratio between testosterone and cortisol and how the EX group was lower than the CON group. In women, the area under the curve of growth hormone and cortisol were greater in the EX group than the CON group. No difference was seen for the area under the curve of testosterone and the TC ratio between the EX group and the CON group. For the EX groups, the area under the curve shows that testosterone, growth hormone, and ratio between T and C is greater in men than women, and the cortisol remains unchanged between the sexes. For the CON group, the area under the curve of testosterone and TC were greater in men than women, and the growth hormone and cortisol between the sexes remain unchanged.
This figure involved the results of the protein expression of PAX7 and showed significant interaction effects of sex versus condition with a corresponding p-value of 0.045. The EX condition showed that there was a greater amount of PAX7 in men than women, as shown in the second two bars in the figure. The CON condition showed no significant difference between men and women, as shown in the first two bars. Looking at just women, women had a PAX7 concentration that was significantly greater in the CON section compared to the EX. However, with the men, there was no significant difference between CON and EX for PAX7 concentration. When looking at CON overall, women had a higher PAX7 concentration than men, as evident in the first two bars in the 12-hour as well as the 24-hour sections of the graph. For the EX sections, there is no significant difference for either men nor women. There was also a significant interaction effect when looking at the time versus condition values of this graph with a p-value of 0.004. In the 24-hour section of this graph, it's evident that the PAX7 concentrations in the CON sections were much larger than the EX conditions as depicted in the first two bars. For the EX conditions, the PAX7 concentration significantly decreased from going from 12 to 24 hours, while having no significant difference from PRE to 12 hours and PRE to 24 hours. For the CON condition, PAX7 concentration significantly increased from PRE to 24 hours, while having no significant difference from PRE to 12 hours and 12 to 24 hours. Figure 4b shows protein expression results for MyoD. This had significant interaction effects for time versus condition versus sex, with a corresponding p-value of 0.027. Within men, looking at the EX conditions, the EX conditions had significantly increased from PRE to 12 hours, while for the CON condition, there was no difference across all times, as evidenced in the first bars in this figure. Within women, the EX condition depicted that MyoD protein content had no change over time, while for the CON condition, the MyoD increased from PRE to 12 hours but decreased from 12 to 24 hours. Figure 4c showed the protein expression results for cyclin D1, which had a significant main effect of time with a p-value of 0.005. This simply showed that cyclin D1 protein expression increased from PRE to 12 hours and decreased from 12 to 24 hours.
Upper body resistance exercise increases in hormones and resulted in greater satellite cell proliferation in men but not in women. They also found that the hormonal response did not affect the increase in satellite cell differentiation. Hormonal response to resistance exercise affects the satellite cell proliferation and differentiation and thus could influence recovery from resistance exercise.
Some plausible mechanisms for the differences in men versus women, as shown in the results, could be due to the effect of estrogen on macrophages, as macrophages have estrogen receptors. Macrophages mediate recovery from muscle damage by inflammatory responses or phagocytosis, followed by anti-inflammatory cytokines to facilitate recovery. The presence of macrophages have been shown to promote satellite cell activation and proliferation, while the absence greatly impairs satellite cell myogenic progression. Another plausible reason is the effect of cortisol, as the different results between sexes could be due to the greater impact of cortisol on satellite cell myogenesis in women compared to men, as they do not produce testosterone, where testosterone has inhibitory effects on cortisol.